The following segment is intended for informational and educational purposes only and was written and developed by BloodStream Media.
In essence, gene therapy is a treatment that's used to fix a disease or a problem that's caused by a gene that doesn't work right. So in the context of hemophilia, the gene for factor VIII or the gene for factor IX isn't functioning properly, and so that protein isn't being produced, or it isn't being produced in a form that functions. So either we introduce a functional copy of the gene, or there are some technologies that potentially could actually correct the defect in the gene. So all of that is gene therapy.
Hi, everybody. That was Dr. Mark Redding, adult hematologist from the University of Minnesota, where he runs the Hemophilia Treatment Center. I asked Dr. Redding, as a physician, why he's so excited to have gene therapy part of the hemophilia treatment landscape.
I think the most exciting thing is that, even though we have really safe, effective treatments now for hemophilia, and we have for a long time, they all still require repeated injections given over a lifetime. And, the newer therapies are subcutaneous, but it's still a sharp, pointy thing. And giving an injection is hard. And none of the current treatments that we have really achieved anything close to what we would consider a cure for hemophilia. And by cure, I mean your clotting ability would be normal enough that you don't need any additional treatment if you injure yourself or if you have a surgical procedure, and a treatment that would ideally be like a one-time treatment that would last you for many years and ideally your whole lifetime. So although we have really good, safe, effective therapies now, we don't have anything that really meets the bar of cure or something close to cure. I don't think gene therapy right now in its current forms is going to be a true cure, but it's a whole lot closer than the stuff we have. So I think it's really about, improving the quality of life with people living with hemophilia, not having to do repeated injections, and then getting those factor levels to be sustained in a range where they don't have to worry about, a bleeding event if they have an injury.
This segment is brought to you by CSL Behring, a global biotherapeutics leader focused on serving the rare disease community by providing innovative therapies. They now have a first-of-its-kind treatment for hemophilia B. To learn more about this treatment option, visit beyondhemb.com and download the B Support app from the App Store or Google Play to stay up to date on relevant information and to manage your treatment journey.
This is a paradigm shift. Instead of having to replace the protein over and over and over again, we're going to take a different approach here, and we're going to have the patient's own liver be the factory that makes the factor IX.
Speaking is Dr. Stephen Pipe, adult hematologist from the University of Michigan.
And so we've had the gene, we've cloned the gene for factor IX, decades ago. That's how we've been making all these recombinant clotting factors from that gene. But that's sitting in a factory somewhere in a big bioreactor making protein. Why don't we get the gene into the liver and have the patient's own liver make the factor IX for them? This, in concept, could be a single treatment event. And then once the gene is there, the normal protein machinery of the cell, of the liver, will be able to make the protein in perpetuity.
So it's really there's a lot of reasons to be excited. I think our excitement has, we've had some ups and downs. Some of the gene therapy studies have run into some problems, and I think this has taken us a whole lot longer than we thought it was going to. I mean, in the late 90s, when I was in a lab doing research as a fellow and right after my fellowship training, back then we thought gene therapy was going to be a clinical reality within 10 years. And here we are, almost 25 years further down the road from that. And it is a clinical reality, but it's taken a whole lot longer than we wanted it to be. So our excitement has been going on for a long time, and we've had some starts and fits, but we're getting there. So this is a very long story with gene therapy for all diseases, and hemophilia has been there from the very beginning.
So Dr. Redding, why is hemophilia such a good match for gene therapy?
Yeah, that's a great question. So it's because hemophilia is caused by a problem in one gene. So there's one gene that doesn't work properly. and because of that one thing, we have the disease or the disorder hemophilia. So we only have to fix one gene. We don't have to fix it all the way, right? So we don't need factor levels to be completely normal to have a significant impact in a positive way on what it's like to live with hemophilia. So I think in the early days we were thinking, geez, if we could just get factor levels, above 5%, that'd be good enough. So not having to correct it fully was appealing. And then, as I mentioned before, the animal models. So having small and larger animal models to study this. I mean, if you don't have a good animal model, it's really hard to develop completely new treatments. It's trying stuff in humans that's never been done before, and the safety issues and all that. So we had animal models that were well described and well characterized. That really helped a lot. But I think the key is that it's a one gene causing the disease situation. There are a lot of other diseases like cancer, for example, where there's multiple different genetic issues that contribute to people developing cancer. And so gene therapy for diseases like that is going to be a lot harder. So hemophilia, because of these things, I mean, I think I mentioned before, hemophilia has been there kind of all along in gene therapy.
Dr. Pipe, with hemogenics for hemophilia B in particular, what are some of the unique aspects of the treatment?
There's a few specific aspects of the hemogenics which I think are really cool. So we got a gift from nature about maybe 10 years ago or so. There were patients in a city in Italy called Padua, and they were getting worked up at the hematologist because they had sky-high factor IX levels and they were getting blood clots. And their factor IX levels were anywhere from 700 to 900%. So almost tenfold higher than the normal concentrations. And when they analyzed those patients, they had a single mutation in their factor IX protein that caused it to be hyperactive or highly active. And so they called this the Padua factor IX. Well, the scientists put two and two together and said, well, the expression we were getting in those previous trials with factor IX, we were getting expression in like the 5 to 10% range. I mean, it was good enough to be a prophylactic therapy, but can we get even higher expression? So the thought was that if we combine the Padua variant inside these AAV transgenes, we might be able to get 8 to 10-fold higher factor expression. So the clinical protocol here was originally developed by a company called Unicure before it was transitioned to CSL Behring. And they already had data with this vector with the wild type, the normal factor IX gene. Those gentlemen were expressing between 5 to 10% levels. They've been followed now for more than 5 years, stable expression. And we wanted to know what would happen if we put the Padua gene in. So at our center and then a couple of other places, we dosed 3 patients with this modified, same vector, but modified with this Padua variant. And those 3 gentlemen got factor IX levels basically up into the normal range of factor IX expression. So that gave us, that really emboldened us that really this is the trick to combine these 2 elements together. So we then launched into what your listeners may know of a phase 3 trial. So all the patients in the phase 3 trial, we enrolled 54 patients into the phase 3 trial. They all had to be on standard of care prophylaxis with factor IX. Then they got a single treatment event with etranacogene, which became hemogenics. And then they were able to stop their prophylaxis. And we followed the patients, and we took a time frame between 7 months post-treatment to 18 months, because that's when everyone had sort of stabilized their expression from their liver. Some of the other benefits that I think I've heard from patients is some of them came into this study with some pretty challenging joints. They still had active synovitis, inflamed joints because of repeated bleeding. And we saw improvements in their joint function, less pain. We saw them taking on new activities that they hadn't been doing previously. And I've caught some of these guys when they've come to clinic for follow-up saying things like, well, when I used to have hemophilia, which is really a really important aspect for me. There's been a mental health change here for the positive. They are now thinking like, I don't think about my hemophilia on a daily basis anymore, or on a weekly basis. I'm not infusing, I'm not getting those regular reminders that I have a missing protein anymore. I'm relying on this prior treatment event, and I don't have to think about my hemophilia. The spontaneity that they report coming into their lives, the spontaneity and freedom that comes into patients' life when they know they've got this steady state expression, it's not changing on a monthly basis or now even on a yearly basis. I think that's a really important aspect of the transformative nature of this kind of treatment.
Until next time, stay curious and embrace the possibilities of a whole new world ahead. Once again, this segment has been brought to you by CSL Behring, which now has a first-of-its-kind hemophilia B treatment. Visit beyondhemb.com or download B Support wherever you get your apps for more information.